Good evening, everybody. I see the room filling up. Welcome to this ESU course and webinar tonight, which is hosted by European Urology and its editorial board. My name, I realize you cannot see me at the moment. They're working on the slides. My name is Maarten Albersen. I'm one of the associate editors. I'm welcoming you on behalf of Jim Catto, who's the editor-in-chief of European Urology, who couldn't make it tonight. And we have tonight a webinar on scientific writing, which will be given by Jim Catto, myself, Sarah Psutka, and Andrea Mari, who's present today. And he's a postdoc fellow in Firenze, working on RCC and urothelial carcinoma. So we'll have 4 talks of about 15 minutes. Jim's is a little bit shorter. It's an introductory talk about the journal. And then we'll take questions after each talk. And if there's no questions, we will shift the questions to the end. And you're being accredited for listening to this webinar. So next slide. This is the overview of the program tonight. First, Jim will give an introductory talk. So it's not a talk about the introduction section of a scientific paper, but it's an introductory talk mainly about the journal, about the impact factor, how you should, what you should or can do to get into the journal. Then I'll highlight methods or give some instructions and tips and tricks for writing your method section. And then it's up to Sarah Psutka from Rochester in the US, who is also one of the associate editors working mainly on oncological papers, who will detail and give you tips and tricks for writing the results. And finally, it's Andrea Mari, again from Firenze, who will discuss the conclusions and the discussion section. So we'll start with Jim's talk. It's quite brief. It takes some questions, and then we'll go over to the next talk. So if you would like to start Jim's video, please. Thank you.
Thank you very much for coming to this European Urology run School of Urology session on how to write a paper. We feel it's really important that you understand how best to write, review and read a paper, and often the best way to do that is to try and do it yourself, try and write and communicate with your colleagues. It's important for many reasons, not least the ability to understand science, to understand medical progress, so you can take forward the best treatment options available for your patients in the future. Over the next hour, we'll try and outline what we think as the editors are the important components of this, and we'll try and convey to you why we think it's important that you understand this. The first thing to say is it is hard. Certainly at European Urology, we receive many papers and accept a small number. We have a very high bar, and that really reflects the quality that we aspire to. However, we have many authors that publish with us for the first time, and many papers were submitted with their fingers crossed, hoping for the best. Overall, our acceptance rate is about 9%. It's been relatively steady year on year, and that again comes back to the quality that we aspire to. But it's not all lost. If you do get through, do get published, you have a paper in a high impact journal that has a wide readership. We have nearly 2 million PDFs downloaded last year, and we have global authorship and readership that we consider ourselves one of the premier surgical journals in the field. So if you do get through, then it's been very worthwhile. We do try and help things. So we've created a family of European Urology journals, including EU Oncology, EU Focus, EU Open Science. Each one has got a different purpose and a different goal. So look on the website and understand which one. You can submit directly to European Urology, and we will read and try out your paper. It'll either get reviewed within the main journal or be allocated to one of the other journals with your permission. If we feel it would sit better in that environment, it would still need peer review there. And again, look on the website to understand what the goals could be. European Urology Oncology is very much about multimodal care for patients, multidisciplinary approaches, and therefore we will look for papers that reflect our purpose and our goal, which is improving outcomes from cancer, whether that be quality or quantity of survival. European Urology Focus is much more syllabus-based, much broader in its drive, much more on a cyclical pattern where we look at each issue will focus on one particular topic, have a number of independent papers that will be related or independent from that topic, but over time we'll build a resource that could be used year on year, time on time. And Christian's done a great job with the journal. We have a good impact factor. As you'll see, it's been revised this year, and going forward we really hope that this will become a stalwart of all our European urology family and our urology reading. Open science is very much about open access, open science, transparent science. So it's about developing a platform for journal whereby papers are submitted. They're all open access, what's called gold open access. And we look at novel formats and novel ways of trying to communicate the message. Very much our youngest member at the moment, but could be one of the most important in the future. We do try and help you. It's really important that you put effort into your paper. As you're going to find over the next hour, we have a clear understanding of how we think papers should be written. We read many a year, so we have a clear view as what is good and what is bad. And it seems it's often quite easy to understand and to see if authors have been sloppy or lack quality. Andrew Vickers has written 2 papers that will be important to you, and they're free to download through the website. The first one is Guidelines of Reporting Figures and Tables. Again, I'm a strong believer on reading a paper from the figures and tables, and that's often the first thing I turn to. Can you understand the patient population? Can you understand the intervention? Can you understand the outcomes? All of these are necessary and avoid duplication. So your figures should not duplicate the text that's written in the results section. Your results should be smooth and transparent and should match the figures without duplication. Same for the tables. Finally, we also have statistical guidelines. So again, look at these. These have been very well read around the world. We had them initially published in European Urology, and they've now been published and adopted across all the urology guidelines, or across all the urology journals. Again, it's important that you understand and report your statistics in a correct fashion. All of the papers that we accept in European Urology undergo statistical review independently, and many of the authors of this paper will be reviewing your papers with that intent. So it pays to listen, read, and understand what they're saying. We do understand about the burden on authors, especially in a journal where we have such a low impact, such a low acceptance rate. So we do allow a first submission where you don't need to do the formatting for European Urology. I would say that you do need to make it look like a proper scientific paper. At the end of the day, we're human, and if we read something that's poorly presented with a lack of focus, too long, not very concise, then I suspect it will always influence our thinking. But you don't need to do all the specific formatting we request. You don't need to submit all the authors at the beginning. If you get through the R1, the initial review, then you'll have to do the second stage, but I'm sure by then you'll be delighted to do so. So thank you for coming along. Thank you for attending. Looking forward to this, and I hope you learn a lot. Please don't hesitate to contact me or any of the editors if you think that'd be helpful.
Okay, I'd like to thank Jim for the talk. Again, if you would have any questions, please use the Q&A box down below. You heard Jim say that we now have a free formatting submission system, which is really a relief for many authors. Still, though, you need to stick to the guidelines of submission, and you need to structure your paper according to what we request. And we'll go over the sections in this webinar. So we'll start with methods, and this is a pre-recorded lecture by me, and then we'll follow the flow of a scientific paper as we go along the webinar. So I'd like to ask Wendy or Marielle to start the talk. Thank you. Hey everybody, thank you for being here tonight, and thank you Jim for the invitation. My name is Maarten Albersen. I'm working in Leuven, Belgium. I'm one of the associate editors for the journal, and I'll detail today on how to write the method section for a research paper. So if you look at the order of sections of the paper that you would typically write, then the method section would be the first that you would write. Why? Well, because you basically wrote it before you started the study. So this section has already been written before you started the study. Basically, it's a rework of the protocol. It also helps you in a way to discover methodological bias at a moment where it's still changeable or easily fixed. Then you write the results, discussion and conclusion, and the introduction is mostly done last. Why is the method section important? Well, one of the main reasons for rejection of a manuscript may be that the reviewers cannot fully understand what you did or how your study was conducted. And this misunderstanding may lead to misinterpretation of the data and may lead to rejection of your paper. So please be aware that the method section is one of the central or the core sections of your paper. It describes how the study was registered, designed, and performed, as well as the way and the statistical analysis that was performed to analyze these data and interpret these data that you provided. It has to provide, therefore, all the elements that allow authors to exactly reproduce your study. And it's often the most concrete and specific part of the text and may therefore be actually the easiest one to write, especially if you have your protocol available. So let's look at the writing style that you would employ for writing your method section. Well, first of all, the complexity of scientific research mandates that the methods are written in a clear and orderly fashion in order to make your writing as reproducible as possible to avoid any confusion and ambiguity. Your writing style should be direct, precise, and in the past tense. So your protocol has already been written. The method section basically or roughly is a conversion of your protocol into the past tense. Section should have already been written, for example, at the time of institutional review board approval or grant approval. Method section is highly factual and therefore, unlike, for example, the introduction or the discussion section, you don't need to take your reader on a path. However, if you organize your method section very well and very neat, that's vital for the reader and the reviewer's understanding. And in most cases, a chronological structure is the easiest to both read and write. So start with general information in your method section that applies to the entire management and then boil down to details of individual experiments that make up the whole story. Match the order in which methods are described with the order of the results. It's easier for the reviewer and easier for the reader if they read a section of the methods that they can scroll down to the similar section of the results if they want to look at a certain specific experiment or part of your research. Include inclusions of the methods that have been described previously and don't go back into describing these in details again. Do not discuss pros and cons of certain methods that you have employed. The method section is not suitable for this. This can be done in the discussion section if you feel that some of these experiments warrant further explanation in terms of choice of methods. So be concise while listing all the details that are vital for reproducibility. The method section shouldn't be overly long. Please be aware that you can use supplementary details or supplementary methods to describe certain protocols and certain experiments into the detail that they actually require if these are lengthy. So what are the contents of the method section? Well, first the research plan, then the materials, patients or the population, and then the methodology that you used. So in terms of research plan or protocol, describe the type of study. So it's very easy for the reader and reviewer to upfront get an idea on what type of study you're handling with. Is it retrospective, prospective? Is it randomized control trial? Also at the start of your method section, include IRB approval and informed consent if needed. And the time frame of enrollment may be important, for example, in diseases which have a rapidly changing standard of care, so that the results and the methods can be interpreted according to the time frame in which the experiments were done. In terms of research planning, please be aware that European Urology and the sister journals require as a prerequisite for consideration for publication that you register your trials, specifically your prospective trials, in public databases such as clinicaltrials.gov and the Prospero database for systematic reviews. On the right-hand side, you can see some examples and excerpts where these registrations are mentioned in the methods section of the respective papers. Please in detail and carefully describe your patient population or your animals in terms of selection criteria and the rationale for including this specific cohort of patients or animals. Exclusion criteria should be described and, if necessary, explained. It is helpful for reviewers and readers if you detail how you chose your control group versus your interventional group. You should report basic demographic data on the study cohort, but this is done in the results section. And sample size can be either included in the patient's description or in the statistical analysis section under power calculations. For basic and translational researcher, please be aware that if you describe your animals, you should include detail on age, strain, gender, weight, physiological or pathophysiological state, maintenance and euthanasia for reproducibility reasons. Identify and describe in detail the equipment and materials that you use. If there can be some variability in production, you should also quote the source, for example, for reagents. Is particularly important. Detail also the origin of biological materials such as cell line, tissues, RNA, DNA. The specific methodological aspects of certain experiments can be briefly referenced if they are standard or have been done before to spare words. If this is not the case, you will need to detail the experiments and the context. The intervention and the endpoints and outcomes need to be clarified. In new experiments, and this is particularly important for basic and translational research, you should always validate your approach and describe the details of the experiment in such detail that they are actually reproducible. And again, you can use supplementary materials for this. If you're in basic or translational science, please be aware that you need to keep a lab notebook to be exactly precise in what you have done in your experiments and give highly detailed info. Methodology is often subcategorized per experiment in basic and translational research, whereas in clinical research, the flow is often an overall overview of the study design, participants, intervention, control, and then statistical analysis. In terms of statistics, again, you need to be precise enough for other researchers to reproduce your finding while not being overly superfluous with giving details. You should include sample size calculation, power analysis, detail on what kind of data you've worked with, what were these normally distributed, yes or no, and how did you test this, and all these kinds of factors are detailed in the guidelines for reporting of statistics by European Urology and several other urological journals. And you can read those in the journal. Also, please, before you start submission of your manuscript, actually basically before you start writing your manuscript, if you want to target one of the European Urology journals, please go to Guidelines for Authors, as you can see here. Being done. You can find them on the website of European Urology, and it's of vital importance to stick to these author guidelines in order to increase your chances of your paper getting accepted. And there also you can find the ESU course presentations that were given today for your reference. The type of study determines the type of reporting that you need to do, and this is also again detailed in the method section. You can refer to the Equator Network for reporting guidelines, and here you can see the example of the consort statement, which is used for the reporting of randomized clinical trials. You can see on the website all the necessary documents are available for download, such as the consort flow diagram that will need to be included as a figure in your randomized trial if you want to have your trial accepted at European Urology. Lastly, before finishing this talk, some tips and tricks. So the bulk of the section of methods should be written before research is done. You should use clean, crisp, direct, and concise language and leave no room for doubt. If you need more detail, refer to the supplementary data and give your detail there. And register your trial and follow guidelines and recommendation on experimental and reporting conduct, which are detailed on the website of European Urology. Before you finish and submit your manuscript or send it to your co-authors, please ask yourself the following questions about your method section to ensure that you have included all the important aspects. Is there sufficient detail for reproducibility? Is there excess information that could be removed without affecting the quality or the interpretation of the results? If so, then please do so to keep the reader and the reviewer's attention. Are all the appropriate controls mentioned and explained, and are all the appropriate citations included? Then for basic and translational research, again, please mention always the sources of your reagents and experimental equipment. So to be very short, how to write your method section, please be concise, precise, and make sure your research is reproducible. Thank you very much for your attention, and I'll be happy to answer some further questions that you may have live. Okay, I see there's no further questions so far. Please again use the Q&A box if you want to ask any questions. There was one question of how do I become a reviewer for the journal for European Urology? Well, first of all, you can register yourself in the editorial manager system, which you can find on the website of European Urology. If you make an account there, you can then register what your field of expertise or field of interest is, and this makes you, well, on the short list, let's say. But basically, we need to get an impression of that you have a specific field of expertise, that we would need your skills and your knowledge to actually evaluate papers. So start writing, start publishing yourself, get involved in the society, for example, via the Young Academic Urologist or via any of the other platforms that the EAU offers. And make sure you get known, make sure you get published, and by that, that your specific expertise is known by the editors, and that you get in that sense invited to review for the journal. So many questions coming in right now. We'll start Sarah's talk. I'll start answering those in the chat box, and we'll answer the rest of those which are the most interesting live after the next talk.
Hi there, my name is Dr. Psutka. I'd like to thank the organizers for including me, and I'm excited to speak with you about advice on how to best present the results of your paper. These are my disclosures. So when you do research, we all know that it's never a straight line. You start with a question, and then as you're doing your analysis, things happen. Data comes up that's unexpected, findings lead you in different directions with your analysis, and ultimately you end up with quite a bit of data that you are in charge of making sense of. However, when you present your results to the reader, you may understand the intricacies of your data, but it's incredibly important that you present a clear, linear discussion and narrative that essentially allows you to walk them down the path so that they can understand what you've discovered and what you think this means and how it changes the literature. Your results is really where this happens. And while the approach to the manuscript in general, in terms of how we present it, is very organized and something that we're all very used to seeing in a specific order, starting with title page and abstract, and then moving through the introduction, methods, results, tables and figures, and then on to the discussion and conclusion. Ultimately, when I write a paper, I take a very different tact. And the reason being that I think it's more efficient to do it in a different way. So I think about categorizing my topics and parts of the paper according to what really flows together. And then I reorganize. So I think that the results and the tables and the figure are really the heart of the matter. And as such, I move them to the very beginning of my writing process. And so this ultimately here is how I structure my papers when I write them. I start by putting together my results and tables and figures. And then from there, I go on to writing the methods, introduction, discussion, conclusions. Of course, your references come with all of that. And then I put together things like my title page and abstract. And why do I do this? Well, the truth is this is a much more efficient way to write a paper. And now everyone writes a little bit differently, but I think that this makes the most sense because if your tables and figures and your results are at the heart of your paper, sometimes you'll find that you actually will write an introduction, go on to present your analysis and put it together, and then realize that the way that you've structured your introduction doesn't really set up the story in as logical a fashion. And likewise, writing the discussion, I think, is really important to do at the very end when everything is organized, said and done, because then you can really use that to efficiently respond to your results, contextualize them within the literature, and then make recommendations for next steps and talk about why they're impactful and meaningful. So let's talk a little bit about anatomy. We're going to talk about the anatomy of those tables and figures, your results text, and then the abstract results. And really, these three things should basically mirror each other. Our minds work in such a way that we are used to seeing data in a specific fashion. It can be jarring for a reader if things are presented in a way that's out of order. And also, it can really help to reiterate a point over and over again, so that by the time your reader completes your paper, they understand not only your key points, but also really understand your main message and also the importance of it. So the first thing you're going to talk about, and as you're putting together your tables and figures, and then ultimately your results, is who is your study about? So figure 1, when I think about figure 1, it's all about the inclusion and exclusion criteria. Now, the important thing is when you're talking about who this study is concerned with, you want to present it in a way that is extremely clear and succinct, but also very transparent. So you'll start with your parent cohort, and then the first figure really shows how you narrowed it down to get to your analytic cohort. And depending on what type of study you are presenting, whether it's a clinical trial, a systematic review, or an observational study that involves, such as a cohort or a case control study, you're going to want to go to the guidelines that are the best reporting practices for that type of study. So for example, if you're reporting a clinical trial out, you'll want to follow the consort guidelines and present a consort figure. Similarly, for a systematic review, you'll follow the PRISMA guidelines and present a PRISMA flow figure. And then if you're looking at presenting an observational study, this is where you would follow the stroke guidelines. And also, I do want to just highlight these guidelines as a very helpful way both to ensure that you are reporting your data at the highest possible standard of reporting, and then also making sure that you're including all of the necessary components. So in addition to pointing you in the direction of these guidelines for the purpose of noting how these figures are formed, I do also want you to look at their key reporting criteria, so that you make sure that you always have those in your papers. The next thing, in addition to telling who your study is about, you want to talk about who those research subjects are. And so while your figure 1 really details your inclusion and exclusion criteria, your table 1 focuses on those clinical, pathologic, and demographic characteristics. Now, depending on the paper that you're writing and depending on the journal to which you're submitting, you may have this in more of a supplemental table form. However, no matter how you write a paper, at some point there will be a table that clearly describes the cohort of patients that you are studying, or the cohort of research subjects. Moving along through the flow of your tables and figures, the next thing you're going to focus on is the exposure variable. So usually my table 2 and my related figures really detail the exposure or the independent variable of interest. And this is where you get into if it was, for example, a specific disease characteristic such as margin positivity, or if it was talking about operative time or distance from a hospital that a patient lives, socioeconomic status or smoking status. This is where you get into things like how frequently patients smoked, their dosing, the amount that they had smoked, how long they've smoked for, whether they quit, when they quit, and so on and so forth. Because this is where you really get into what the factor that you're trying to understand its association with your outcomes. This is where you get into the details that you collected on that specific factor. And then your subsequent tables really get into your analyses. So this is where you detail the response or dependent variable, and then you get into your univariable or unadjusted analyses, and then ultimately your adjusted analyses, and then any sensitivity analyses, validity assessments, and so on and so forth. So there's going to be, in terms of my key points, the first thing, everything has to be presented in a logical order. It is key that each figure or table must have a legend, and the legend should follow in the text of the manuscript. In general, I like to present these on a separate page. Once I've completed my manuscript and the references, I include a page in my Word document that is my legends of tables and figures, so that the reviewers clearly have those details for them. Your legend should be clear and succinct, but detailed. They should tell the story of that figure, so that if your reader only saw that figure, they could interpret the data there. Every abbreviation should be explicitly spelled out. This is something that goes without saying, but oftentimes is forgotten just as we get busy writing these papers. And I do think it's important that when possible, think visually about your tables and figures. You want your color coding to be clear. You want to synthesize and highlight key findings in these figures, and we're going to kind of get into some even more specific recommendations next. So the first question you want to ask yourself at every point when you're putting a figure in your paper is, does this figure help my reader understand my findings more clearly? So here's a bar chart. that shows the volumes of patients or the proportions of patients in a study with different stages of lung cancer. This is not terribly helpful because you could simply include a table and the data would actually be a little bit easier to understand and a little bit easier to interpret. The reader can go straight to those proportions, and all of a sudden that data is much clearer, whereas the bar chart takes up a lot of space and doesn't actually synthesize any additional findings. Similarly, think visually. It's very common for authors to present studies that, or present figures that utilize standard different formatting from, for example, Excel or whichever spreadsheet program it is that you're making your graphs in. But for example, while these 3D figures may look kind of neat, they don't really help you interpret the data, and to some degree, they actually make it harder to read. For example, the x-axis is challenging to read. They don't necessarily add any additional data, and it's not necessarily that easy for you to figure out, for example, what these changes even mean. For example, this is a much more streamlined way of presenting this data, which is looking at changes in kidney function scores between two different groups across age variables. So these scatter plots with lines of best fit are much easier for a reader to kind of look at, note the differences in the lines of fit, and interpret. Again, so we want to only include a figure if it improves your reader's ability to understand the findings, and avoid selecting a graph from pre-selected options just because it's easy. Think about the data and think about how you can best interpret that data visually so that your reader will take home the point almost immediately. Make sure you don't rely on a graph to replace key numbers. If there's a point that you need to make in terms of numbers that you want reported, it's critical that you include those in the text. The reader should not have to hunt for the important details. And then make it beautiful. I think that's something that Andrew Vickers says in this guidelines for reporting of figures and tables from his paper here. And I think it's really important because it can be hard when you're looking at a table such as this, which is looking at differences in outcomes in 2 clinical trials across 2 different regions. One can look at this and see a bar graph with 2 different colors, but one can't really note the substantial or sort of interpret the differences between the 2 groups. This is a much more efficient way. It's horizontally laid out to interpret this data and to present it in a way that allows the reader to see that there is a discrete difference in both the regions between the 2 treatment arms, the red and the blue. And it's just a little bit easier visually and succinctly to put it for the reader to interpret this data. Again, in terms of making it beautiful, colors, graphs like this that have lots of colors, lots of lines, and where it's really almost important to understand sort of the scatter plot looking at age versus total PSA at treatment. This is really hard for readers to understand and takes a lot of time to sort of look at and suss through. Here, on the other hand, is an example of a really visually attractive graph that's also very easy to understand, where you can clearly see the distributions of intact to free PSA separately by cohort. And notice that there's a clear difference between the top cohort and the others. So again, label your axes. Make sure to report percentages rather than proportions, just because you're thinking about how do our minds work and what's going to be the easiest way for your reader to take in the data you're presenting. Be consistent across figures in terms of colors and font. It's just more visually pleasing, but it also looks cleaner, and it looks like you put a little bit more time into things. It's important to add your distributions to your line plots, and certainly add the numbers at risk to your Kaplan-Myer curves. These are 2 Kaplan-Myer curves that actually are kind of challenging to understand. One of them is just the straight statistical output that's been represented. The other is challenging to read because of all of the sensor lines, whereas this KM curve is much cleaner and easier to understand. It's color-coded, and you can clearly see the number at risk in the appropriate color coding. Finally, I think there are some recommendations from Dr. Vickers' guidelines that are worth taking home. Avoid including, for example, the receiver operating curves. Just include the area under the curve. It's much cleaner and requires much less space. And in general, avoid graphs that don't, again, synthesize or add any additional data to your paper. So avoid things like pie charts, simple bar graphs, and 3D charts that have a low data to ink ratio. Just use a table when you're reporting these numbers, but use visual depictions of data when it helps you to synthesize points. Avoid excessive white space and be mindful of the tails that can really skew the data. If there's not a lot of data in your the ends of your Kaplan-Myer curves, truncate the follow-up if the number at risk is low. And then when you're thinking about your graphs, put them on light backgrounds just because they're easier and cleaner to interpret. And then I'm going to say it again because it's often forgotten, include a descriptive title and a legend. Your tables similarly should be clean, clear, and aligned, simply presented with consistent fonts, avoiding redundancy. And so, for example, if you have a binary variable, instead of presenting the proportions as you do here, as you see here for men and women, you can simply report the proportion of women because you know that the men is the converse of that. So that limits the number of lines in your table and makes it a little bit easier. And you can see here how I've justified these different columns in a way that is visually pleasing and also a little bit easier to understand. I think it's tempting to include a lot of p-values when you write these papers, but you really only want to report p-values for analyses for which you have a specific hypothesis and be very clear about what that hypothesis is. If you have a categorical, multi-level variable, only include the one test for that unless there's a reason to look at the p-values across different levels. Avoid, again, repetition and empty cells. Ensure you include the number of patients and the missing data so that the reader can understand why your proportions may not add up to 100 if they're assuming that your N is your total N for your study. And when possible, for example, as you see here, condense categories when it's meaningful. I think there's a few other things that are important to do. One, you want to be very clear about what statistics you're reporting and make sure you keep them consistent within a table so that the reader understands, are you looking at comparisons between medians and interquartile ranges or means and standard deviations? Always include your units, and definitely when you're presenting multivariable models, ensure that you clarify the reference level so that your reader understands what those point estimates are in reference to. Small points, but try not to avoid the plus or minus. Use things like 95% confidence intervals or standard deviations, which are much cleaner and more appropriate, and avoid long tables. When you look at big tables like this, it's really hard for readers to look through and identify the key information they're supposed to come away with. And your eyes just kind of gloss over these sorts of things. This is a much cleaner depiction of that data that we just looked at, which came from a systematic review that colleagues and I just recently wrote, that makes it clear which categories within a specific assessment scale were, which criteria were met by different studies, as opposed to, as if we go back, just a number of zeros and ones. And then I know we've spent a lot of time on tables and figures, but once you have those all done, the writing is easy. Because at this point, all you're going to do is use your results section to walk your reader through your tables and figures. So paragraph 1 is your cohort discussion. And you're going to reference those first tables and figures that described your inclusion and exclusion criteria and who those patients were. Paragraph 2 is the details about that figure, that table 2, your exposure variable. Paragraph 3 describes your response variable, your primary outcome of interest, and then your unadjusted and adjusted analyses. And then your paragraphs 4, 5, and 6 are going to walk your reader through key analyses, validity, and the sensitivity analyses. For the abstract, you're going to mirror this and simply change and summarize those paragraphs into single sentences. And that's going to be the abstract results section, which you're going to cleanly write at the very end once you have everything else done. So to start to summarize, make sure you have clear callouts here. You should have your tables and figures each enumerated within your text. Your results should be logically organized, and you're always going to be specifying which hypotheses you test. Report both your statistically significant and insignificant results because you want to build credibility and tell your reader that you're presenting all of the relevant information here, what you found mattered and what you found didn't. And then just a really important point, if you described an analysis and the methods, make sure you present the results. And if you describe a result, make sure you presented the methods that allowed you to arrive at that result. So just to wrap up, again, your results section, your tables and figures should be clear, succinct, with the appropriate level of detail to help you spin your narrative. Show your data in a way that tells the story of your research and lead that reader down the garden path in a clear way where they come through the end of the results section, completely understanding the key findings of your study. And make it easy for your reader and your reviewer. This will not only help your reader to understand your data better, but also will be helpful in the review process. Thanks again to the organizers for including me, and I'll pass it off to our next speaker.
Thank you, Sarah, for an excellent talk. Again, there's some themes that come back. So enough detail, not overly detailed. Be clear, be precise. So it's more or less the same in methods and the results section. There have been some questions that have been answered in the box. You can find them down below at Q&A. We'll go to Andrea's talk, who will discuss the discussion and the conclusion section. You can keep asking your questions. We will answer them through the Q&A box, or if any major questions, we'll take them at the end of the session live. So Andrea, thank you for being here, and the screen in this case is yours.
Hi, Martin. Good morning, everybody. Can you see my slides?
Yes.
Okay, perfect. First of all, I would like to thank Professor Palou and Professor Liapsikos for the kind invitation, and Professor Catto for the organization of this UROwebinar. Is really a great honor for me presenting how to write discussion and conclusion in this webinar. So briefly, this is my conflict of interest. Surprisingly, I will start from conclusion and not from discussion. Why? For me, conclusion probably is one of the most difficult paragraphs to write, and one of the less satisfying, of course. Why? When you are writing your articles, it takes so much time for you to make all the projects, to analyze data, and then to write that in the conclusion section, you should summarize the most important results of the article in a very, very few sentences, underlining all the regional aspects, and that is not simple at all. Also, conclusion is one of the first read section of a manuscript, because usually the readers start with the abstract. Of course, they read the aims of the study at the end of introduction, then they move to the results and take a look to the figures, to the tables, and then probably they jump from discussion to the conclusion section. They usually read the conclusion, and probably, basing on the conclusion, they will decide to read or not your manuscript in detail. So this is very important. And for this reason, conclusion should be effective and attention-grabbing. So my personal advice is don't write the conclusion after discussion, and especially if you are tired. This is not only my opinion. Of course, there is an article published on a PLOS journal in a survey conducted on early career researchers that show that nearly 40% of them found the conclusion one of the most challenging part of a manuscript to be written. Conclusion has several formats. I just bring you some examples. In BJU International, for example, there is not a special paragraph for conclusion, and conclusion is just the last period of the discussion section. On the right side, you can see a conclusion section of a paper published in European Urology, and it's the same journals, and conclusion is considered separately from discussion section. So it's very important to read the journal guidelines. Usually there are not many information about the length of conclusion, but just try to take care to avoid to bore the readers. So these are my personal tips and tricks. Conclusion should be justified by the results that are reported in your manuscript, so without any further hypothesis. And through these chapters, show to the readers the quality of your work, its novelty, the importance, and afterwards also consider to read with attention the journal's guidelines before sending the article to the journal. Don't introduce new information that are not previously written in the results and the discussion section. And conclusion, of course, is not a mini abstract of your paper. So you cannot only summarize your findings. Try to don't be redundant, and in the conclusion section, you shouldn't look to the future projects, but just stay on your findings and wrap them up. So let's move forward to the discussion section. Also, discussion, of course, is very important. It's extremely difficult to write, also as a conclusion, and it allows the authors to make sense of the reported data. That means that they have the possibility to show the rationale of the work and the stream of talks conducted during the setup of the study. Discussion also allows to contextualize the results in the light of the current literature and topic. That means also compare the findings with those reported by other studies. And of course, consider also the consequences on the clinical practice and their implications. In discussion, we have to summarize the main findings. You can underline the strengths of the study, but also you have to explain the methodology and its limitation. This allows you to report the pending issues and finally take a look to future projects. There are few general rules in discussion, but discussion should start answering the initial question. The initial question is generally postulated in the introduction, generally in the second chapter of the introduction, introducing the topic, or when explaining the aim of the study. Then, in the intermediate section, authors should summarize the main findings, then interpret your results and explain the differences of our results with those reported in other studies, underlining, if possible, the novelty of our results. This point can be repeated according to the number of the most important findings in our study. That means that if we have 3 very important points, you have to repeat the period 2 or 3 times, and in each period you start to summarize the main point, you interpret these results, and then you can explain the difference. Place your finding into the field. That means where do we stand now with clinical practice and where are we going? It is very important, and also understand if the study may improve the standard clinical care. Also consider if your study can pave the way to future studies. Of course, discussion cannot be always the same. There were some questions about this topic, also about methods. It's exactly the same for discussion. I just bring you an example: meta-analysis and systematic review. In meta-analysis, the authors usually analyze possible outcome, and if it's possible, and if it is positive or negative. So the authors can discuss the reason of these results. They provide an interpretation and comment on the characteristics of the studies included in the review, and especially on the experimental design that the authors use in each study. Usually, meta-analyses are written by a panel of experts on a specific topic, so personal opinion should be avoided. Finally, meta-analysis usually synthesize the existing evidence from multiple studies. The authors, relying on their findings, should recommend key suggestions for conducting and reporting future primary studies. What about the length of a discussion? Generally, the length of discussion should not exceed the sum of introduction, material and methods, and results. That means that discussion should be around 6 or 7 paragraphs, 200 words each. So it should be important to count repeatedly the periods that you are writing. You can do it when you are writing the first draft of this discussion. Probably you will do it if you are very experienced, or otherwise you can do it afterwards. But this point is very important to try to not avoid the readers. And the discussion section is usually divided in three separate paragraphs. An introduction paragraph, as I told you before, about the rationale of the study and the topic introduced. An intermediate paragraph where the results are summarized, then are interpreted, and then are compared with the studies presented in literature. Finally, a conclusion paragraph acknowledging for the limitation of the study, from the strengths, and then considering all the future studies that can be written after your study. About the discussion, some tips and tricks. Each word of the paragraph should be counted and placed carefully. That means that each word whose removal will not change the meaning should be taken out probably from the text. Another point is that in discussion, you don't have to mention all the literature, all the studies about a certain topic. This is almost impossible. As you know, most of the journals have at least 30 references possible, and this the author should try to concentrate on those studies that are most connected to our findings. Another point is try to avoid discrepancy between all the references. So I will show now an anatomy of a discussion through an example. This is a multi-center retrospective study published in European Urology from Professor Shariat, and it's about the role of pathologic nodal staging score in patients treated with radical cystectomy for bladder cancer. In particular, the authors propose a diffusion tool for the use of adjuvant therapy in this setting. In this case, the discussion is very didactic, and the discussion on the right side this is the first period, again answering the initial question. On the left side, the introduction was speaking about the importance of nodal disease as a powerful predictor of oncologic outcomes and the importance of lymph node dissection. And again, in the first sentence of the discussion, the authors wrote that lymph node status is a powerful predictor of bladder cancer recurrence and survival after radical cystectomy. In the following sentence, again, they repeat the importance of a lymph node dissection and the number of lymph nodes to better stage the disease. In the second part of the discussion, the authors analyze and summarize their results. They interpret and then they compare with available studies. This point is repeated more than one time. In this particular case, in this article, the authors use many times the pronoun we. We will see also the next slides. So the authors found the accurate prediction of real lymph node stage based on pathological tumor stage, LVI, and number of lymph node examined using the NSS is feasible. Then they interpret the results, and afterwards they compare with available studies. The third period of our discussion, again, the authors they make a summary of another important point of their study about lymphovascular invasion. They summarize the finding according to the tumor stage. They interpret this result and they compare again with available study. In the fourth period, again, they summarize another result and they interpret. in this case, there is no comparison with other studies. So this is the last period. The authors acknowledge for study limitations. So you can see the first limitation is the study design, multi-center, retrospective. A second limitation that is that the authors can control for treatment decision, and in particular patient and surgeon preference, and they don't have information about the anatomic template of lymph node dissection. The third limitation, that central pathology was not included. And the last sentence is about the strength. Another possible additional sentence can be about the importance of future studies on this topic. Anyway, the authors did not mention it. So these are my final suggestions. Discussion should answer the main questions that is raised in the introduction. Discussion should always be supported by the results, and discussion, of course, allows the interpretation of the results, their critical appraisal, and their evaluation in the context of available evidence and current literature. Finally, conclusion should be informative, effective, and supported by your results. So I really thank you for your attention. If you have any question, please write to us, and I have concluded. Thank you.
Thank you, Andrea. Very nice presentation, very comprehensive, very clear. The way we should write papers, basically. If anybody has further questions for Andrea or for me, please hit us up in the chat box. I think there were a couple of questions asked that may be interesting for you to go over again, mainly pertaining to reviews, systematic reviews. What should I do to write a good introduction section and a discussion section in the area where evidence is lacking? So we try to answer these to our best of our abilities. If there's any questions, hit us up. Furthermore, I suspect there's quite a lot of people listening who are in early stage of their career. Further tips, general, besides the specific sections of a paper, please find yourself a mentor who can help you write, practice, make schemes before you start writing an article. You can find these presentations on the website of European Urology. I think Sarah gave you a schedule on how to structure your article. I gave you some tips on how to structure your article. So make post-its or make a schedule, then start writing. Write more words in the beginning and just scrap away all the extra info that you feel that that is doubled or that you don't really need. And really work with somebody who has experience in writing. And you will notice that the first paper that you write will come back completely red with all things crossed and marked and changed. And it's a frustrating process at sometimes, but you can grow very fast into becoming a good scientific writer, I think. Andrea, any further comments from your side?
No, I was answering to a question about the statistics methods and the platform we use for statistics. Of course, is another very difficult point of writing a paper, preparing a manuscript. Probably I will suggest you to talk with a statistician. If you are working in a university hospital, probably it is more simple for you. Otherwise, just try to search on Google. There are a lot of information today about statistics. The most difficult part probably is coding, because we haven't studied so much statistics, at least in my university. So I had to start again with everything, and it wasn't simple, of course. I found some statistician mentors that they helped me, of course, a lot, but probably the first thing that you should do is try to find the solution by your own way, and then try to talk with somebody that probably will help you. But today we have really a lot of information on the internet. Probably you should know how to interpret all the informations, because when you are coding, you just put a code and then it runs, but you don't really know if your result is correct or not. So try to discuss it with your mentors and with a statistician if it's possible, but try to start doing things by yourself and then discuss with other person that have more experience than you.
I agree. I think trying for yourself is really a good learning experience, but always test what you did with somebody who really knows and is experienced in statistics, either your mentor, a senior person in your research team, or your local statistician can also help you a lot. I know my current PhD student, he learned R from YouTube a lot. He took also a crash course, and now he's really proficient with it. So there's a lot of ways to learn. A lot of universities have local courses as well. There's one question pertaining on what should you do if you report on a lab-based experiment and there's no comparing studies. So what you should do in the methods is really make clear what research that you did, make sure it's reproducible. Also, in the case that it hasn't been done before, make sure that you validate your model, for example, yourself. Don't assume that the model you use is a good model, just validate it before you go to interventional experiments, for example. And then in discussion, there's always ways to report on what you found and to place it into context. If there's no context, if there's no context that you can write about, then really the question is whether it's worthwhile to have done the experiment. So you can place them always into some kind of clinical context, or what your findings would mean when you would translate them into clinics. Let's see, another question is, can the platform EU provide us with the courses in this direction? For example, SPSS, I don't think so. I think we do have statisticians that we work together with, like Andrew Vickers, who's a very big help together with his team in the journal, who wrote us also the Statistical Guidelines. But I do think for learning statistics, it's you're basically best helped at your with your local statistician. I'm not sure. You can always ask the people of the School of Urology whether there's some kind of course, like an introduction to statistics or survival statistics or clinical statistics that are used a lot in urological publications, but I don't think the EU offers it at this moment. Let's see. Lessons on calculating, entering results, data collection, statistical interpretation. Again, I think that's the same. Well, this is the question by the same person. So I think we're done. No more open questions. Andrea, any last comments?
I think that, well, just the last point, SPSS is a good platform because it looks simple. Anyway, when you are doing statistics on SPSS, it's extremely difficult to remember all the processes that you have. And this can introduce some errors that you are not able to find afterwards. While in R, when you are coding, you have all your coding that's saved. And also, when you are publishing in very high impact journals, you can share with the reviewers your coding, and probably it's the best way to do statistics.
Yeah, at European, we actually ask that you submit code. Yeah, you're not obliged to, but we do ask to do so.
Okay, I have no other further comments.
Good. I think we're done. I would like to also thank Jim and Sarah for the presentations. Please know that you can find the presentations online, both at the UROWeb website at the section of the school and the webinars, but also at the European Urology site, as well as with the talks that were given in the same webinar the years before. So thank you all for your attention, and enjoy EAU next week, and hope to speak to all of you soon. Thank you very much.
Thank you. Goodbye.